This is my project 1.2 plan after video. So ATP, what is it? It is adenosine triphosphate. It is basically the cell's energy, and ATP is released when the terminal phosphate bond is broken. Yeah. Anabolic pathways. Anabolic pathways consume energy to build complex molecules. There are generally dehydration synthesis reactions. Yeah. Catabolic pathways. They release energy by breaking down complex molecules into smaller compounds, and they are generally hydrolysis reactions. Yep. Metabolic pathways. Okay, so in a metabolic pathway, it begins with a... What was this? I know this. It begins with a specific molecule and ends with a product where each step is catalyzed by a specific enzyme. Yeah. Define what metabolism is. Metabolism is the totality of an organism's chemical reaction. Yeah. What is extension? This is the I know this. This is the end step of in a PCR reaction, where the thermostable DNA polymerase synthesizes a new DNA strand. So basically makes it's a replication phase. Yeah. What is found in PCR reactions? Ooh, PCR reactions. There's 6 things in there. DNA nucleotides, primers, thermostable DNA, template DNA, the last 2, I forgot. PCR, buffer, and magnesium. Okay. What is denaturation? Denaturation is the first step in a PCR cycle. It is when something's heated and where hydrogen bonds between the bases, like the A and T and G, are disrupted, which the product is like a single strand DNA molecule. Yeah. Annealing. Oh, that's the second stage of a PCR reaction, and it's where PCR primers are made of DNA nucleotides and not RNA. Yeah. What is a cloning vector? Oh, I know this. The cloning vector is the original plasmid in gene cloning. Yeah. What is the polymerase chain reaction, PCR? Basically, PCR is it is a type of it's the process where it produces many DNA sequences and target DNA sequences, specific DNA sequences, and then there's 3 steps. There's heating, denaturation, and then annealing, and then extension, which is replication. And when this happens, there is a exponential replication of identical DNA molecules. Yeah. DNA ligase. DNA ligase is the enzyme that seals the bond between restriction fragments. Yeah. Restriction enzymes. They cut DNA molecules at specific sequences, and those sequences are called restriction sites, and They're also known as restriction endonucleases. Yeah. Well, that's it.